introduction. Good morning, everyone. My name is Monica Baciu. I'm PhD student in Professor's Berenwinkel Group at ETH in Zurich. And I'm here today to present oncotree, a method for embedding and clustering of tumor mutation trees. And first, I'd like to introduce what mutation trees are, how they capture the evolutionary history of the tumor, and more specifically, how they model the accumulation of driver mutations over time. And by mutations here, I mean all kinds of genomic, epigenomic events, or even changes in gene expression. And in this figure, we can observe the accumulation of driver mutation that give rise to different cell subpopulations, which are called subclones. But in reality, what we observe is only the state of the tumor at the time of the biopsy. So what is not observed, the history of the tumor in the past, can be inferred using mutation tree inference methods. They start from the sequencing data at the time of the biopsy, and they identify the subclones that share the same genetic profile. And based on the cell fractions in each subclone, it's able to infer the temporal order of the mutations. So the tree representation encodes that. We see the nodes that stand for the subclones, and their labels are the driver mutation labels. And the structure of the tree tells the temporal order of the accumulation of driver mutations. And for the purpose of our project, we will discard the subclone sizes and the branch lengths. And from a computational point of view, the mutation tree is just a labeled tree, where the labels are numerical encodings of the events that happened in each subclone. And for simplicity, we will use colors to represent the labeled nodes. So why the sequence of mutations is important? We can have two types of sequences of mutations. So co-occurrences, that means accumulations of mutations in each subclone, or mutually exclusive pairs of mutations that occurred independently in distinct subclones. And this leads to a branching pattern in the mutation tree. So there is literature showing that the order in which the driver mutation occurred is very important and leads to different clinical outcomes. So for example, there is this famous paper that tells that the group of patients that acquired mutation in TET2 first and JAK2 later had a better survival than the other group that had the reverse order of the mutations in the two genes. So given that, and given a cohort of many mutation trees that come from different biopsies, our goal is to find groups of tumors with similar evolutionary history. And here, oncotree2vec comes into play, and it learns vector representations, embeddings, for each mutation tree. So our model learns embeddings that capture the different relationships between the three subclones, and the matching trees will have a close representation of the embedding in the embedding space. And there are several node relations that we want to encode in our embedding. And here is a list of the ones that we take into consideration. So first of all, we consider neighborhoods of different sizes around every node in the mutation tree. So here is an example of neighborhoods around the start node of the mutation tree. Neighborhood of size 0 means the individual node, and we expand the neighborhood such that a neighborhood of size 3 around the start node covers the whole tree. In addition, we take into consideration pairwise relationships between the nodes. And these are biologically relevant. So these are the root-child relations, the direct edges, the co-occurrences, and the mutually exclusive nodes in the mutation tree. In addition, we take into consideration substructures of the mutation tree by discarding their labels. So this helps us model the different branching sizes of the trees. And for that, we take into consideration the same type of neighborhoods as I mentioned before, but this time we discard the labels. So each of these node relations can be represented programmatically as a concatenation of the numerical labels of the nodes. And this can be seen as a vocabulary of words. So here on the slide, you see the vocabulary of node relations for two mutation trees. And what is matching between the two trees is highlighted with colors. So not only the individual nodes are matching, the ones with labels 5, 6, and 7, but also the node relations between nodes with labels 6 and 7 are matching. So here we have a direct edge, and here we have a co-occurrence in the tree B. Also, we have a matching mutual exclusive relation between labels 5 and 6. And also some tree substructures are matching, namely a linear pattern of 3 nodes. So we discard the labels, we just look at the subtree structures, and we see that the sequence of 3 nodes is common between the two trees in this example. So, as I said, we can view each mutation tree as a vocabulary of words. And now, how do we turn this into a tree embedding? And here's the idea. We use a natural language processing model for document embeddings. And such a model exists. It's called doc2vec. And its input is texts that belong to documents, and the output is embeddings that are encoding those documents. And it's not a black box, it's a simple neural network that learns to predict if a word is in the context of a target document. And one of the header layers is actually the embedding of the document. So given that, we have a model, oncotree2vec, that learns the embeddings for the mutation trees and is able to put together in the embedding space the trees that are similar. And using these embeddings, now we can put our method into a framework that is used for clustering mutation trees. So we perform hierarchical clustering using the cosine distance to compute the similarity between embeddings. And thanks to the embeddings, we can visualize the tree cohort using UMaps, or so here using the heat map to visualize the clustering. And now I will show you 2 applications on 2 real data sets that we use to assess the performance of our method and the usefulness of our method on real data. So the first data set is a cohort of 123 AML mutation trees. And by clustering them using the oncotree2vec embeddings, we found 13 large clusters that are governed by the primary gene mutation. So here, what was mainly matching between the trees was the root-child relation. And this confirms the literature, because for AML, the clinical protocol says that the primary gene mutation is the strongest signal and is the first criterion for diagnosis. Further, we applied survival analysis on the groups that we found, and the result was that we found two groups with a significant difference in survival. So I'm talking about the first group is cluster 1, where mutation IDH2 was governing the group. And this group had a very good survival. And on the opposite side, the other group consists of clusters 4, 9, 6, and 3, with the indicated primary mutations in each group. Of course, as you can see on the heat map, there are many other smaller clusters of 2-3 patients that have strong matches between the node relations, other than the root-child relations. And those matches indeed are confirmed in the literature as being known pathways for AML, but we couldn't drive any other statistics because the groups, these groups are very small in this cohort. The second experiment we had was to apply oncotree2vec on a cohort of 43 trees from 6 cancer types. And for this cohort, we were only interested in the tree structures, so we discarded the labels, because we wanted to see the different modes of evolution. So we wanted to see if the clusters given by the clustering the embeddings would highlight the different modes of evolution. And this is indeed what happened. We found 4 clusters. Each of the cluster contained trees with a different mode of evolution, linear punctuated branching, and the 4th cluster had linear to branching evolution. And these modes of evolution match the corresponding cancer types in literature. So for example, it's known that liquid biopsies, like we have in AML and mesothelioma, have a linear pattern. Then we have punctuated evolution for uveal melanoma. For breast cancer, we have branching evolution. And these results are confirmed in the literature. So, of course, in the paper, we show many other experiments, comparisons to other in-use distance metrics, and we assess the stability of the learned embeddings among multiple runs. We also have a discussion about the fact that we provide the user the ability to weight the different categories in the node relations vocabulary. So for example, for the second experiment, we weighted more the subtrees without the labels, and we set weight 0 to the labeled relationships, because here we were interested in a specific pattern. So yeah, by weighting the different vocabulary categories and by guiding the user to do that, we can highlight more stronger patterns in the data. So the paper is available on bioinformatics, of course. And in conclusion, the embeddings learned by oncotree2vec are valuable because they can be used in different contexts. So you can use them to compute a distance score and use it for clustering. Then having the embeddings rather than the distance scores makes it very easy to visualize the cohort in a lower dimensional space, and makes it easier to use the trees in a downstream analysis by using their embeddings. Also, oncotree2vec overcomes the problems of other in-use tree distance metrics. And more specifically, we account for biologically relevant node relations. We saw all kinds of node relations that are encoded in the embedding. Then we don't have the infinite sites assumption. That means our method is applicable for copy number trees, for example, which is not the case for most of the distance metrics in the literature. So all in all, it's an unsupervised learning method, and it works for any cohort size, which is not the case for the deep learning methods in general. So with that, I want to thank you for your attention. I want to acknowledge my group in Switzerland, especially Professor Baciu-Dragan, who is my advisor, and all the other members of our group who are very helpful in brainstormings and feedback. And also I want to thank my family for all the support. Thank you, and I'm waiting for your questions.
Amazing work. Questions?
Hi, thank you for that. I'm sorry if I missed something, but I'd like to ask you to clarify how you're utilizing the structure of the trees themselves. So the hierarchical relationships and so on that are implicit there.
Yeah, so maybe this slide is helpful. You have here a concrete example of how we encode the tree structure. So again, we start from every node, but without the labels. So say you start from this node, and then you have, so you start from a node, and the neighbors are root, node, node, right? And then you encode this in a string, and this is the word that encodes this substructure. And you enlarge the neighborhoods. So here was a neighborhood of size 1 that I showed. And a neighborhood of size 2 will be will cover the whole tree. And
okay, so in the embeddings, then you're leveraging the order of the labels in that case.
So these are the words, right? That will be converted. All of them, all the relations will be encoded in the embedding.
Okay, so that gives, yeah, got it.
Yes.
Thank you.
Hi, thanks for interesting talk. The AML results are really interesting. I think the recovery of the IDH gene is really like very promising. Have you done or future plan for further validation using the subtype AML subtype annotation or treatment outcome data? Cross-checking your clusters with those?
So for this specific AML data set, we did a survival analysis. So for the clinical data, we only look at survival, which was the thing people are interested in. But of course, one can investigate more of the clinical data to see matching patterns.
Okay, thanks.
And of course, okay, this AML data set might not be the best one to put value on the method, right? But in the literature, there are many other public tree cohorts that we can apply the method on, but we only included in the paper a few experiments also because of the lack of space.
Yeah, if you can further link this to the treatment outcome or the subtype, which is very closely related to the also the treatment outcome of the AML patients, will be really interesting.
Yep, sure. Thank you.
Really cool method. I'm excited to try this on my data. Do you have a sense of like the sample size you'd need for something like this? I see you have like a large vocabulary. Does it matter if, 50, 20, or do you need, I know you did 100.
So of course, if you have 4 trees, the input to the neural network is much larger because each tree is translated into the corresponding vocabulary of words. But this method works regardless of your cohort size, because what it does actually is just, okay, so it starts with random embeddings, right? It's an iterative process, like in a neural network training. So it starts with random embeddings, and in every iteration, it takes pairs of points, pairs of embeddings, and they are pushed apart if the distance between them is high, like there are many differences between them. Differences mean the intersection of the words is very low. But then if the trees have many words in common, then they are pushed together in the embedding space, right? And so you do this iteratively. So regardless of how many trees you have, even if you have 2 trees, the embeddings will be learned accordingly. So that's the advantage of this method, that you're not constrained on the number of input, the size of the tree cohort, the number of input trees that you have.
Great.
It's
thank
by
you.
design.
Yeah.
Yes.
Great.
Also have a quick question. So you're converting your trees to sentences, essentially, to text, and you use document
Exactly.
embeddings. Have you considered using graph embeddings, like for example with graph neural networks? Because a tree is a graph.
So this is a much simpler method, architecture of the network than graph neural networks. And so there's no need to try something more complex because this works and this is interpretable and everything. But as I saw in the literature, graph neural networks are usually used in the proteomics. For example, you have the protein-protein interactions, and you are able to learn embeddings for every node.
Also for the graph.
And then you combine the node embeddings to get an embedding for the graph.
Right.
So of course you can use that as well, but it's more complex and there's no need for that in this particular problem.
Interesting. Okay. Thank you. Okay, let's thank the speaker.